The overall goal of this multiplexed immunofluorescence analysis is to automatically count cells based on their phenotype.
This method allows multifluorescence staining and automated cell counting to analyze the phenotype and function of tumor and immune cells within a tumor microenvironment. This technology facilitates the generation of new type of composite prognosis or predictive biomarkers derived directly from a tumor microenvironment.
Generally, individuals new to this method may struggle, because the phenotyping procedure is very long and the resulting raw data requires further processing. We first had the idea for this method when we attempting to characterize the phenotype and immunological interactions of intratumoral T cells.
After thawing, use a paper towel to carefully dry the slides around the tissue sections and encircle the tissues with a hydrophobic barrier pen. When the barrier has dried, fix the samples in 100% acetone for 5 minutes, followed by 2 minutes of air drying. Then wash the slides in TBS for 10 minutes. Pre-treat the slides with 3 drops of 0.1% Avidin for 10 minutes in the dark. Then tap or flick the slides to cover the tissue with the Avidin solution. Add 3 drops of 0.01% biotin to each sample, and tap or flick the biotin across the samples, followed by a TBS wash as just demonstrated. Then block any non-specific binding with 100 microliters of 5% normal serum in TBS for 30 minutes at room temperature. At the end of the incubation,